Assalamu alaikum. Today I discuss histology of the respiratory system. It consists of lungs and a series of air passages and its functions the inspiration of the oxygen from the environment and expel the carbon dioxide to environment by the process of expiration. It has structural classification and functional classification. First is the structural classification. This is upper respiratory tract up to larynx, and this is the lower respiratory tract. Start from trachea to alveoli. Functionally, it divides into two parts, conducting part, respiratory part. The conducting part is include the upper respiratory tract and also part of the lower respiratory tract, trachea, and the primary bronchi up to terminal bronchiole. So, end of the conducting system is the from nose to terminal bronchiole. Respiratory portions is start by the respiratory bronchiole, then alveolar duct, alveolar sac, and alveoli. What is bronchial T? It is a system of airway that allow passage of air into the lungs where gases exchange occur. These airway are located within the lungs. So, this is trachea, this is extrapulmonary bronchus, this is interpulmonary bronchus, and gradually the cartilage is in the trachea is continuous and C-shaped, and this cartilage became irregular and ultimately cartilage is disappear. When cartilage disappear, it's known as the bronchiole. This is terminal bronchiole up to terminal bronchiole. This is up to this conducting part, and in conducting part no gaseous exchange is occur. Respiratory bronchiole start the respiratory portion of the respiratory system. Here gaseous exchange is occur. So what is the function of the conducting system? Conducting system function air conduction, air filtration, and function of the respiratory system is gaseous exchange. In addition to the air passes through the larynx, produce speech, and olfactory mucosa of the nasal cavity carry sense of smell. So these are the extrapulmonary region and interpulmonary region of the bronchial tree, and this is the respiratory portion. The mouth, nose, nasal cavity, the function of this part of the system is to warm, filter and moisten the incoming air. The pharynx here, the throat is divided into trachea, windpipe and esophagus. There is also a small flap of cartilage called the epiglottis, and this epiglottis is close the larynx during when food passes from the oral cavity to esophagus. Larynx also known as the voice box. This is the vocal cord. By using uh, expelled air, we can produce sound. Trachea is also called the windpipe. This is the tip where they carry air from the throat into the lung. The bronchi, the trachea divided into two tip called the right and left principal bronchi, which enter within the lung. The right principal bronchus is shorter, um, wider, and more vertical. So frequently foreign body is lost in the right bronchiole. The left principal bronchus is longer, narrower and longer and more oblique. So foreign body is not usually impact within it. The bronchiole contain irregular ring of the cartilage, and next portion of the bronchial T is the bronchioles, less than 1 millimeter in diameter, and there is a very narrow tube structure. There is no cartilage. For support, a continuous layer of smooth muscle cell is present. Alveolus is the thickest thin wall which permit the exchange of the gases, oxygen and carbon dioxide. This is olfactory mucosa present in the um, roof of the nasal cavity, and it's contain bipolar neuron, the supporting cell, basal cell and Bowman's gland. The respiratory epithelium is a specialized pseudostratified ciliated columnar epithelium that line most of the respiratory tract, including the nasal cavity, trachea, bronchi. It play a crucial role in air filtration, humidification, and protection. These are the ciliated cell. This is goblet cell. This is the scanning electron microscopic picture showing the cilia, and in between cilia these are the apical portion of the goblet cell, and this is the mucus secretion from a goblet cell. The respiratory epithelium consists of 5 types of cells. The most numerous, most common cell are the ciliated cell, and its function move mucus and particles upward for clearance. Location toward the respiratory tract. This is goblet cell. Its basal portion is narrow, but apical portion is expanded with its secretion. It secretes mucus, and within the mucus, dust and microorganism is trapped, and with the cilia, these particles is removed from the respiratory tract. This is basal shape. It is act as a stem cell. It's located in the basal region, and it can convert it into any type of cell of the respiratory epithelium. Then the brush cell. Brush cell function detects irritation with the sensory microvilli, and club cell. The club cell or pleura cell secretes surfactant and detoxify harmful substance. Location bronchioles. The endocrine cell secrete hormone and neurotransmitter and secreted in the respiratory tract. Now the histology of the trachea. It is a short flexible air tube about 2.5 centimeter in diameter and 10 centimeter long. It's from within outward. It has following layer: mucosa, submucosa, cartilaginous layer with trachealis muscle and adventitia. So this is the trachea. This is the C-shaped tracheal ring. Here you see the two-piece. No, this is not two-piece. This is a continuous C-shaped hyaline cartilage present within the trachea, and this gap is present posteriorly for easy food passes through the esophagus, because the esophagus is related posterior part of the trachea. And this is the trachealic muscle, smooth muscle. It can contract. When contraction occur, the diameter of the trachea is reduced. When relaxed, diameter is increased. This is the mucosa, and this is showing the lining epithelium, lamina propria, and the now the so this is the mucosa, the lining epithelium, pseudostratified ciliated columnar epithelium. You see the nucleus is not single layer. It's look like multiple layer and leave a pinkish ribbon like area above. So it is obviously pseudostratified ciliated columnar epithelium. The next layer is the lamina propria, and then next layer is the submucosa, which contain dense irregular connective tissue, but lamina propria is the loose connective tissue. So dense irregular connective tissue from submucosa, which contains ceramicus gland, white are mucus gland, and darkly stained are the serous gland. This is trachealis muscle, and this is formed by the flattened nuclei with flattened cell, squamous cell. This is smooth muscle. This is the hyaline cartilage. You see the chondrocytes and matrix is homogeneous due to the presence of collagen fibril. Collagen fiber is absent here, present collagen fibrils. This make the cartilaginous matrix homogeneous. This is perichondrium, the outer connective tissue covering of the cartilage. This is adventitia. It contains dense irregular collagen-rich connective tissue, contain blood vessels, nerves, and um, adipocyte. So this is another histological figure of the trachea. This is pseudostratified columnar epithelium. You see the nucleus are multilayered and leave a pinkish area. And this is lamina propria. This is submucosa contains seromycus gland. This is mucus acini. This is serous acini. And this is the lacunae contain chondrocyte. This is glass shaped homogeneous cartilaginous matrix. This is perichondrium. For drawing, you can use this type of picture. Now the bronchus, the right and left principal bronchus, and it has two parts, the extrapulmonary part and interpulmonary part within the lungs. There is some similarity between trachea and extrapulmonary bronchus. The C-shaped cartilage present within the trachea, but irregular cartilage plate present within the extrapulmonary bronchus. Tracheal is smooth muscle cell present posteriorly, but smooth muscle cell is surround the wall. The diameter of the trachea is larger than the extrapulmonary bronchus. The secondary bronchus or interpulmonary bronchus, mucosa, pseudostratified ciliated columnar with goblet cell, laminar propria, and muscularis mucosa of the smooth muscle fiber, and submucosa contains seromycus gland, and irregular plate of the hyaline cartilage is present, adventitia is present. So this is the mucosa is formed by the pseudostratified ciliated columnar epithelium. This is lamina propria, loose connective tissue, and there is a surrounding a muscle layer is present. This is the unique features of it. The interpulmonary bronchus, it is absent in and trachea. So you see there is a cartilage is present, but cartilage is not continuous. It is continuous cartilage is present, and this is surrounding adventitia is present. Bronchiole. The structure of the bronchiole mucosa lined by the simple cubital epithelium with pleura cell, lamina propria content, muscle layer is well developed because for support there is no cartilage is available in bronchiole. When cartilage is absent, the structure is known as the bronchiole. So terminal bronchiole, there is no cartilage, so muscle layer is well developed and adventitia is present as usual. So in terminal bronchiole, you can remember three: no cartilage, no glands, no goblet cell. So this is the structure of the terminal bronchiole. You see, this is the simple cubital epithelium. This is lamina propria, and this is well developed smooth muscle cell here, and this is adventitia. So in bronchiole, this is another picture of the bronchiole, terminal bronchiole. Respiratory portion, respiratory bronchiole. Respiratory bronchiole also contain mucosa, simple cubital epithelium with few scleral cell and alveolar branching from the wall. The wall of the respiratory bronchiole is not continuous. It interrupted by the alveoli bulge, and that is why gaseous exchange is possible in respiratory bronchiole, though is lined by the simple cubital epithelium. Respiratory bronchiole is more or less look like terminal bronchiole, but uh, this outpocketing of the alveoli absent in the terminal bronchiole. So let's see how respiratory bronchiole is look like. You see, this is the cubital epithelium, then thin layer of the lamina propria and um, muscle layer, but muscle layer is not continuous because um, the structure respiratory bronchiole, the structure is not continuous. There's a out pocketing of the alveoli is present here, so gases exchange start from the respiratory bronchiole, and this portion is the alveolar tract, and this is alveoli. Alveolar duct possesses no wall of their own. They all long straight tube lined by the simple squamous epithelium display numerous outpocketing of the alveoli uh, and in the alveolar sac. Alveolar sac are composed of group of alveoli clustered around a common air base. Alveolar septa. The wall between adjacent alveoli is separated by a septa. This is known as the alveolar septa. It is formed by the two layers of the alveolar epithelium with their basement membrane and capillaries. So, alveolar, this is alveolar septa, and there in between, within the alveolar septa, there is a gap is present. This is known as the alveolar pores, which allow equalization of the pressure between alveoli, provide a collateral air circulation. So, this is the alveolar pore, this is the uh, alveolar septa, and this is the alveoli. So alveolar septa is formed by the in two sides there is a simple squamous epithelium at middle. This is the basement membrane and blood vessels. This is the structure of the alveoli. Three type of cell is present here. Type 2 alveolar pneumocyte and type 1 alveolar pneumocyte and alveolar macrophage is present here. This is the alveolar macrophage or dust cell. So this is the type 1 pneumocyte, this is type 2 pneumocyte. Gaseous exchange is occur through the type 1 pneumocyte. Type 1 pneumocyte is occupy most of the surface area of the alveoli, but its number is less. Type 2 pneumocyte is occupy less surface area, but its number is more, and it secretes surfactant which reduce surface tension, so alveoli is not collapsed during expiration. And this is alveolar macrophage, so any dust particles is removed by this alveolar macrophage. You see the shape of the nucleus, kidney shape, and its surface is more or less irregular. So this is histological picture of the lung. This is interpulmonary bronchus. You may or may not be found this structure within the lung tissue. You may find it, you may not find it. And this is the terminal bronchiole. It's easily recognized by its well developed muscle layer. This is respiratory bronchiole. This is alveolar duct, and this is alveolar sac, and this is alveoli. Surfactant. Secretion of the surfactant by the type 2 cell. Surfactant is a protein lipid complex. So this is the type 2 cell, and this is the rough endoplasmic reticulum, which um, secrete essential materials for the surfactant, which pass through the Golgi, and from the Golgi the surfactant is produced and it's packed within a um, secretory vesicle, and its shape is unique, is lamellar body layer structure is present here, and it's accumulated in the apical region. When it needed, it by the process of exocytosis, it exit the cell membrane and secrete within the alveoli. It is a mixture of a phospholipid and a protein which reduces surface tension within the lung, secreted by the type 2 alveolar cell. It is stored and transported by the lamellar body. This is the lamellar body. There's a layer structure is present here, and fetal surfactant produce begin around 24 to 28 week of the pregnancy, but it is not fully mature until the 34 to 36 week. Premature infant often lack sufficient surfactant, leading to the respiratory distress syndrome. This is the respiratory system. Baby born before the 34 week of the gestation often lack enough surfactant that help reduce surface tension in alveoli, keep them open. So this is all about respiratory um, system, histology of the respiratory system. And at last, there is the most important portion, the respiratory membrane, where gaseous exchange is occur. After that, our video will be end. If you um, find this video helpful, please share this video. And now let's discuss about the respiratory membrane. It is this thin layer of the surfactant is present here. Then type 1 pneumocyte and basement membrane of the type 1 pneumocyte, and then the basement membrane of the endothelium, and lastly endothelium. So this is the respiratory membrane. This layer have to pass oxygen and carbon dioxide. So you see the deoxygenated blood came through the pulmonary artery, and this um, carbon dioxide rich blood, the carbon dioxide is entered within the alveolar cavity, and the here air is present, oxygenated air is present here. So from this air, oxygen is entered across the respiratory membrane, and it's attached with the RBC. So this is the respiratory membrane, and this membrane is to the main function of the respiratory system. So this is the special figure, and he's showing the respiratory membrane. This is the endothelium with RBC, and this is the type 1 pneumocyte, and this is the type 2 pneumocyte, and this is the macrophage. And here gaseous exchange is showing by this arrow. Oxygen enter and carbon dioxide out. So this is type 1 pneumocyte, cytoplasm, basement membrane of type 1 pneumocyte, basement membrane of type basement membrane of the endothelium. So this is all about histology of the respiratory system. If you find this video helpful, please share this video. Thank you for watching.